sample preparation. You can use petri dishes for cells or glass cover slips, but I suggest you to buy cover slips or petri dishes which are designed for TIRF microscopy. Samples can be cells or proteins, and here you don't have any restriction for the fluorophores that you can use in your experiment. In some techniques, you need special fluorophores like phototransformable fluorophores. Here you can use any. The thing is that, of course, the fluorophore has to fit your microscope, so to the excitation and to the filter set for excitation and also for detecting the emission. You don't need also special buffer conditions such as you would need in single molecular localization microscopy techniques. Of course, you want to introduce some oxygen scavenger system to prevent or reduce photobleaching. It's practical sometimes to create this kind of flow cell. So here you have a cover slip, two cover slips which are separated by something that can be a double-sided tape, for example. So you can inject your sample, and if you think you want to change the conditions in your sample, so you can simply change the conditions by manually, or you can couple this system to a microfluidic device which allows you to change the conditions faster and more precisely. One important thing is that you want to investigate that should be close to the cover slip. So you can investigate, for example, adherent cells, but if your cells are not adherent, you should do a special surface chemistry that can be based on the use of polyal-lysine, collagen, or fibronectin. You can also use the PLL, the polyal-lysine, polyethylene glycol, and the streptomidine biotin system that is actually used in the micropatterning-based applications that I will discuss a little bit later. Okay? And so you want to have a properly aligned laser beam, no optical imperfections. Actually, I don't think that you can see here due to the contrast, but if you have optical imperfections, sometimes you can see interference fringes on your image. So if you see these things, then you have to think about to check your optics. Okay. So let's see first some applications from the protein biochemistry, which is related what we do in PAGE. So our main interest is the study of the different cytoskeletal polymer networks, including actin and the microtubule filament systems. And just to show you again, so actually the development of the TIRF microscopy boosted this cytoskeletal polymer research because it gave a lot of flexibility to investigate these polymers under different conditions. Again, if you compare the wide field illumination of actin filaments with the TIRF illumination, you clearly have much better resolution, much lower background to see individual cytoskeletal polymers. Okay. So for those who are not familiar or not on the cytoskeletal polymer field, these two animations from the inner life of the cell shows the actin, the two main constituents of the cytoskeletal filament networks, the actin filaments, which are assembled from subunits called actin monomers. You can see here, and these are the microtubules which are assembled from the alpha-beta tubulin heterodimer into this ion rod. Okay, so if you focus on actin, so actin subunits can polymerize or assemble into these higher order structures, and that process is strictly regulated inside the cell by a huge number of regulatory proteins. And this slide, what this slide emphasizes is that different proteins can have different activities. That's one thing. The other thing is that they can act the same time, so the activities in a simpler way can be simply added up, but they can be combined in a synergic or antagonistic fashion, which makes it sometimes difficult if you want to do protein biochemistry and you want to understand what is happening in your sample. Okay? So there is a fluorescence spectroscopy approach to study actin assembly that's based on the use of a fluorophore, that's pyrene iodacetamide. So pyrene is coupled to the actin subunit to the G-actin, and when the actin subunits incorporate into the filament, the F-actin, the fluorescence emission of the pyrene increases due to the increase in the quantum efficiency of the fluorophore. So you can use this approach to study the assembly kinetics of actin. And actually the assembly kinetics is affected by many different parameters, such as the concentration of G-actin, the concentration of filament ends, as well as the association and the dissociation rate constant of the subunits to and from the filament ends. Okay? So if you have your protein and you put in this assay, maybe you can see these very strange kinetics, which is sometimes difficult to interpret and completely understand what is going on. However, when the TIRF was introduced, the actin and also the microtubule, the researchers working on actin and microtubules started to use this technique because of actually the obvious advantage of the technique that you can see the individual filament. So from their number, their shape, and so on, you can get already a lot of indication for the activity of the protein that you are studying. Okay, so in this movie, you can see individual actin filaments which are growing on the surface. The surface is treated specially to attract actin filaments in the evanescent field. So you can study not only the structure features of the system, but also the dynamics of the system. So for example, if what you can do is to look at an individual filament, and by relatively simple image analysis, you can generate this kind of chymographs, and the analysis of the chymographs, for example, the slope here will give you the growth rate of the polymer. If you change your condition, you can also have a polymer shrinkage. You can, in a similar manner, you can quantify the shrinkage of the polymers. You can measure also the number of the polymers, which can be indicative for different activities, and also the structural features. So here, for example, branched actin filaments can be seen. That is a characteristic feature of the actin-related protein complex, the R2-3 complex, and here an actin bundle can be seen, which is formed by assembling many individual actin filaments. So these bundles are much thicker than the individual actin filaments. Yeah. So related to your research, we are interested in the regulation of the neural cytoskeleton, including the actin and the microtubule cytoskeleton. This is just to introduce briefly. A nerve cell can be seen here schematically, and if you focus here to the growth cone, different protrusive structures are formed, including the lamellipodium, which is an actin-rich structure, as well as these finger-like protrusions, the filopodia. In these structures, both the actin and the microtubule cytoskeleton have an important role, as well as the associated proteins. So one of our protein of interest is called the DOM protein, the additional associated activator of morphogenesis. And this is a forming protein, and actually it turned out that it's important in the formation of axonal filopodias. So this work was done by our collaborator, József Mihály, from the Biological Research Center from the other side of the river. Here you can see primary neurons, this green fluorescence shows the actin-rich filopodia, and if in the absence of this forming, the DOM mutant, the number of the filopodia were significantly decreased, and that is what we wanted to understand, how this protein can contribute to filopodia formation. And yeah, just to briefly go through, so this protein that is a multi-domain protein. It contains two conserved regions, the FH2, the forming homology 2, and the forming homology 1 region. It also contains an interesting C-terminal region as well as N-terminal elements. And when we started to investigate it, so this is just the standard confocal microscopy. What we have noticed that when we overexpress only this FH2 domain, then we don't see any filopodia formation. However, if we overexpress both the FH1 and the FH2 region, then we see a marked increase in filopodia formation. So to start to investigate the molecular mechanisms, we did TIRF microscopy experiments to study actin assembly. And the first movie will show a standard control. The control, so the spontaneous growth of the actin filaments in the evanescent field, and that can be quantified. And in the presence of these two regions, the FH2 and the FH1, FH2 regions, if you compare these two movies by the first one, what you can notice that there are much more actin filaments. So the filament number is significantly higher. That is indicative for an activity that we call nucleation activity, but the length of the filament or the growth rate of the filaments are significantly smaller, and that told us that these two regions can enhance the nucleation but inhibit filament elongation. In another, that is just another movie that was done by Tom Pollard from another lab. Actually, they did TIRF microscopy. Here, the green fibers are the actin filaments growing, and this red dot is a forming, which was labeled by a quantum dot. So what you can note here that the formins can track the barbed end or the growing end of the actin filaments continuously while the filament elongates, and that is what we call a processive elongator. That's a special class of actin regulatory proteins. Okay. So to understand what can cause the difference in the cells between the effects of the FH2 and the FH1, FH2 regions, we performed the dual color TIRF microscopy. Here we used another protein, profilin, which is an actin monomer binding protein, to see how the profilin can influence the activities of the FH2 and the FH1 regions. So here the experimental setup was that first we used red actin and formed these red actin seeds, and in this experimental setup we used the flow cell. So after a certain time period we changed the solutions to green actin filaments. So what we can see is then the growth of the green actin monomers, or the association of the green actin monomers to the filament end. And in this way we can specifically study the elongation of the actin filament. So that's again just a control which is quantified here. In the presence of the FH2 domain, actually we could observe the same thing in terms of elongation. So the filament elongation was almost completely blocked by these regions. However, to our surprise, in the presence of the FH1, FH2 domains, the filament can elongate at the same rate as the control filaments. So these experiments told us that profilin is a molecular switch in the function or in the activity of the FH1, FH2 domain. Actually, profilin binds to the FH2 region that tunes the activity of the FH2 domain. Okay. And one of our recent study focuses on the C-terminal part of the protein, which is this one. What we have noticed, again in TIRF microscopy analysis, here we quantified the number of actin filaments filaments formed under different conditions. So this is the control, and for different in the presence of different constructs. So that's in the presence of FH1, FH2, and that is in the presence of a construct that contains the C-terminal elements as well. And what you can see just by looking at the images, that here we have significantly more filaments than in the case of the C-terminal lacking construct, and that was actually quantified. So we see a significant increase in filament number. Interestingly, the C-terminal region alone is not able to induce an increase in the filament number. That's again quantified here. So this tells us that this C-terminal region can affect the activities of the FH1 and FH2 regions. We are currently investigating the molecular mechanism, so far I cannot explain you what is happening. Okay, and another thing what we started recently, and that's based on TIRF microscopy as well, is to study the actin and the microtubule crosstalk. And yeah, so using TIRF microscopy, we can simultaneously visualize individual actin filaments and microtubules labeled by two fluorophores with two different spectral properties. So this first images corresponds to the actin filaments, while the second panel corresponds to the microtubules. And interestingly, what we found, this is the control where the actin filaments and the microtubules, hopefully you can see on these merged images, are physically separated, so there are no connections between them. And what we found, that this previous construct of the DOM that contains both forming homology domains as well as the C-terminal region can physically link the actin filaments and the microtubules. So if you look at this merged image, these white regions are indicated for the co-localization of these two polymer networks. So we are currently trying to develop an assay, because these are fixed samples, so the dynamics in these samples cannot be really seen. What we are currently investigating or developing is an assay in which both actin and microtubule dynamics can be followed in real time in TIRF to study how this then forming can mediate the crosstalk between these cytoskeletal elements. Okay, and now let's go to the micropatterning. Which I think is also a powerful technique. If you combine with TIRF, I'm going to show you a few examples. Okay, so here a micropatterning means that we generate a pattern on the surface with different geometries, and for that, this is one of the principles that can be used. So first of all, this is your cover slip, and then you make a PLL PEG coating, polyallyzing polyethylene glycol coating on this surface, and that is a cell or protein repulsive layer, this green one. And then you design your pattern on the surface of the geometry that you want to induce to the surface, and you use this chromium mask here. So you put the chromium mask on the PLL PEG coating and illuminate the whole thing with a deep UV light. And the deep UV light, so this chromium mask is transparent here where you see the white regions, while it's not transparent for the UV light where you see the brown regions. So in this way you can destroy the PLL PEG coating here where you have the white regions that would correspond to your the geometry, the protein or cell attractive geometry that what you want to have on your surface. And then you can functionalize, so since the PLL PEG coating is destroyed in certain areas, you can functionalize these areas with something that can specifically attach your cells or your proteins. And with that you can define or generate a well-defined geometry. to your surface, and since TIRF is a near field imaging technique, you can image what is happening here. Okay. So to show you an example, I take a few movies that were made by in Laurent Blanchoin's lab. So one thing is that you can do any geometry that you can imagine on the surface. So you can generate this circular geometry here. That's not my first movie. Check. So using the micro-patterning method, you can generate this geometry, these radial bars, the circular pattern, and you can functionalize these regions with a protein. In this case, these regions will functionalize with a protein that will attract the actin filaments. So here the fluorescence comes from actin filaments. And in this study, what was investigated, how the geometry influences the structure of the higher order structure of the actin filaments. And the main conclusion was that the geometry of this pattern can govern the higher order structure of the actin filaments, because at these low angles, close to the center of the circle, the actin filaments were parallel to each other. It means, as is schematically shown here, that they different and face towards the opposite direction. So a plus end here, a minus end here, while at larger angles, that corresponds to the blue filaments here, that was the anti-parallel. At larger angles, the filaments are oriented in a parallel fashion. So the same ends face towards the same direction. And one thing that was a second paper, that if you add now myosin to your system, and that is shown here, these different filament networks or subpopulations, the parallel and the anti-parallel, will contract differently. That actually makes sense if you consider that the myosin, in this case that was myosin 2, in the minus end of the filament, and that requires anti-parallel filaments. So first, this inner ring will collapse and the parallel filaments will remain. Okay. Yeah. Another thing is that was done by one of my previous colleagues, is the reconstitution of the actomyosin-dependent mechanosensitive behavior of the tailin-vinculin complex at the focal adhesions. So that's also a cell-free study that uses TIRF and the combination of the micro-patterning approach. So here you can see schematically the principles of micro-patterning. So these spots were functionalized by actually tailing the focal adhesion proteins, and all the other regions are protein repellent, which contains still the PLL-PEG coating. And one thing that was shown is that tailing, using fluorescent labeled vinculin, that vinculin will bind to the tailing functionalized spots only if actin and myosin present together. Okay? And the other thing, which is shown here, hopefully the contrast is enough. So here, maybe you can see these circles. These are the tailing functionalized circles. The green corresponds to vinculin, and there is also actually a red fluorescence that corresponds to the actin-myosin contractile bundles. And what we can see, or what we could see here, if you had a better contrast, is that the force generated by the actomyosin bundle that is attached to this functionalized spot will result in the detachment of the bundle from this spot, and that detachment is followed by the dissociation of vinculin. So these are the chymographs. So this region corresponds where the actomyosin bundle is still attached to the spot, but after detachment, the fluorescence of vinculin also decreases. Okay? And actually that led to this, the working model that kind of explains the mechanosensitive if there is no force, then the vinculin cannot bind to the tailin because the binding sites are not accessible. However, if the tailin binds to the contractile ectomyosin system that can exert force, this force will pull on the tailin, which results in a conformational change, so the vinculin binding sites are available, vinculin can bind and attach to the tailin functionalized spots, which mimic actually the focal adhesions. And when the ectomyosin bundle is released, then also the force kind of disappears, and that directs tailin to its original conformation, so vinculin will dissociate as well. Okay. Okay. And now let's go to some cellular studies that also highlight some important features of the evanescent field. So here what we can see is the dynamics of the microtubule cytoskeletal imaged by total internal reflection fluorescence microscopy. So this is an epifluorescence image, and that was made by TIRF microscopy. The image is inverted. Okay? And if you look at the dynamics, so these are individual microtubules. If you look at the dynamics, what you can, I guess it's easily notice that the intensity is inhomogeneous along the microtubules. So if you look at the tip of the microtubule is much darker, and since it's an inverted image, that would be much brighter in the original fluorescence microscopy image. Actually, here you can see a detailed analysis. The artificially coded image shows the homogeneity along the microtubule. So here white corresponds to high intensity, blue corresponds to lower intensities. And actually this intensity difference along microtubules, and since this movie was made by TIRF, was interpreted as microtubules are going into the evanescent field closer and closer to the cell membrane, to the boundary of the cell, and that's why actually the intensity increases along the microtubules, because if you go inside the evanescent field, the intensity will increase exponentially towards the boundary of the two surfaces. And why I show this movie, because actually this highlights that if you do TIRF microscopy and you want to quantify intensity, that you have to be careful, because if molecules are located in different regions of the evanescent field with different intensities due to the evanescent decay, they can, even if you have a single molecule, you can have different intensities. So if a molecule is closer to the boundary, that will give you a higher intensity than another one which is farther from the boundary. So the intensity in this way is not an absolute measure of the amount of molecules. Another one is still located to studying the ventral regions of the cytoskeleton inside the cell. So this is a confocal movie, not a TIRF movie, that just highlights the initial observations. So here, in principle, red fluorescence emission will show you the actin cytoskeleton, while this green fluorescence emission corresponds to fascin, which is tagged by GFP. So fascin is located into these finger-like structures called filopodia. And what can be observed in these movies, that filopodia are growing and then they fold back. Start again. Be here and here as well. So they will fold back towards the cell and actually incorporate into the lamellar cytoskeleton. And fascin also dissociate from these actin bundles. And to investigate in details the process, the epifluorescence and the TIRF imaging was combined on this movie. So here again red, which is hardly visible, at least from this position, corresponds to the epifluorescence. What what you get from the sample, while the green will show the TIRF, the fluorescence emission due to the TIRF illumination. Maybe on these still images you can see better. It's actually what is happening, that before associating to the lamellopodial networks, these philopodia will go into the ventral regions of the cell. So initially, if we look at the still images, initially the philopodia are red, here for example, and here, and as they fold back they become green, and that means that they enter the epifluorescent region of the cell that corresponds to the ventral regions before they incorporate into the lamellopodial network. Okay, and yeah, at the end I would like to briefly introduce another application related to TIRF microscopy. That's, these applications are based on single molecule localization microscopy, the STORM, the stochastic optical reconstruction microscopy, and the PALM, photoactivated localization microscopy. Okay. So this can be combined with TIRF, and the TIRF technique will contribute to these techniques with its own advantage, to be able to selectively illuminate a region, a thin region, a few hundred nanometers thin region close to the surface. Okay. So actually, TIRF microscopy was used for single molecule imaging quite early. So this is a study from 1995, and that was the setup for the TIRF microscope. And what was imaged is the ATP's activity of myosin. So the glass surface was functionalized by myosin. These spots show individual myosin molecules, and the nucleotide was coupled with another fluorescent dye. And actually, as the nucleotide binds to the myosin and releases to the myosin, then this, the nucleotide spot will appear on the myosin spot or disappear from the myosin spot. So if you simultaneously image these two fluorescence channels, or this you will image the appearance and the disappearance of the spot, you can analyze the kinetics of the ATP turnover of myosin. Okay. Yeah. So I would like to just briefly go through the principles of single molecule localization microscopy. So if you have been in Paris, you might have a chance to watch the Eiffel Tower in the night, when you can see light bulbs continuously from the tower, but every hour for a limited period of time, what you will see that these bulbs are blinking. Okay, so you can see single individual bulbs. And actually, if you now take photos of each at each time point, on your photos you would see different light bulbs, but you would see definitely separately the different light bulbs. And then you take your photos and locate the light bulbs on each photo, each light bulb. You will have many points with many coordinates, but you would know the exact location of each light bulb on the tower. And from this information you can build, reconstruct the image of the Eiffel Tower. Actually, briefly and simply, that's the principle of single molecule localization techniques. One thing with these techniques, that these are required a special fluorophores called phototransformable fluorophores, which can be either photoactivated or photoconvertible fluorophores. The photoactivable fluorophores, to simply introduce the principle, initially the fluorophore is inactive. So first of all, you need an activation beam that will activate the fluorescence of the fluorophore, and once the fluorescence is activated, then you can shine light on that and you will get the fluorescence emission. Actually, this activation will change, as you can see on these emission and absorption spectrum will change the spectral properties of the fluorophore. And again, schematically, what you are doing in single molecule localization microscopy. So first of all, your fluorophores are not activated, so everything is dark. And then you use the activation beam to activate a small number of fluorophores. Then you use the readout beam to illuminate these fluorophores, image them, and locate them, XY coordinates. Okay? And either you photo bleach them or you illuminate for a long time, so they will automatically photo bleach. So you destroy these fluorophores, these cannot be illuminated anymore. And then you start your cycle again. Again, activate certain fluorophores and read out. You will read out these fluorophores, localize the fluorophores. Okay? And here the localization, to localize molecules, the image of individual fluorophores are fit with a Gaussian function, and from this fit you can find the x and the y coordinates of each fluorophore. One important thing that here the resolution, what limits the resolution is how many photons you can capture from a single fluorophore. So the more photons you can capture, the better the resolution you can have. Yeah, but definitely you can have better resolution than using TIRF microscopy, for example. Here you can have a few tens of nanometer resolution. Okay, and again, just a kind of textbook image to highlight. If you look at your sample, I think it's a wide field microscope, and if you look at your sample with a single molecule localization microscopy such as STORM, you can clearly see more details, spots which corresponds to individual fluorophores. Actually, in collaboration with Miklós Erdei from the Department of Optics and Quantum Electronics, we already tried to image actin filaments, single actin filaments using STORM. So the TIRF combined with STORM technique. So this is a TIRF image of an actin filament and a small actin nuclei. And if you do the STORM, you can have localize the individual filament, individual fluorophores along the filament and around the filament, so you can get here again a better resolution than that would be provided by TIRF alone. Okay, and actually you can have axial information in these single molecule localization techniques, such as with STORM. If you induce it doesn't work anymore. If you induce a cylindrical lens, then the image of the object or the shape, or precisely the ellipticity of the image will be sensitive to the depth. As you can see on the middle matrix in Z, so depending on the depth, it can have more circular or more elliptic shape, and from that shape you can have an indication about the depth. So you can actually acquire three-dimensional images with a TIRF, more three-dimensional images with a TIRF STORM combination. And just one example at the end from the literature. So here the cytoskeletal structures in a neural cell were imaged. And one thing that was revealed that the dendrites and the axons had different actin organization, which is maybe, if you look here, so in the axons, that's cross-section, you can see this circular pattern formed of short actin filaments, and this pattern is also highlighted along the axon. So it's a very repetitive circular pattern, while in the dendrites there are more long-acting filament. And actually that's the scheme, how we can imagine these circular repetitive patterns. So these green things are small-acting filaments which are linked together by adenosine and also the spectrin tetramers. Okay? So that structure previously previously was not resolved by other fluorescence microscopy techniques. Just one thing, so here the periodicity of this pattern is around 200 nanometer, a little bit below, like 180 or 190 nanometer, the periodicity. And actually what I mentioned you, the structured illumination microscopy can improve the resolution of the conventional light microscope. And actually we try to see this again in collaboration with József Mihály. We try to see this pattern in neurons using a different technique, and hopefully maybe it's the most pronounced is here. So you can see these stripes, this is again the actin cytoskeleton in the neurons that appear similarly, and the periodicity that we get using the structured illumination microscopy is very similar, that can be derived from the storm image. Okay? Yeah. So at the end, to kind of sum up all these things, so the TIRF microscope is a near-field fluorescence microscopy technique which provides us a surface selective illumination due to the evanescent field. It provides resolution improvement in its own in the axial direction to a few hundreds of nanometers. That depends on the properties of the evanescent field. It's a very useful tool in cell biology if you want to image processes near or close to the cell membrane, and also in protein biochemistry study, if you can design and have a proper surface chemistry that can attach your system here to the surface, and also it can be adapted to and combined with other techniques such as single molecule localization microscopy. Okay. So this is the homepage of our group, so if you are interested in what we are doing, you can visit it, and that's my group. Here I would mention on the we did the DOM studies on actin dynamics, and Peter Gassner, he's an undergraduate student who is actually working now on the actin microtubule crosstalk. And also previously I worked in the CNRS in Gypsy-Rivet, where we had also a TIRF system, and one part of the work that I showed you here was done there. Okay, so thank you for your attention, and I'm happy to answer if you have any questions.